Everyone. So it's really an enormous pleasure to introduce Aviv Regev as our second keynote speaker of this meeting. I had to realize when you're organizing this conference, you actually have very little or no power because you completely depend on these abstracts that come in. So the only thing you can actually do that we as three organizers could do is pick a couple of keynote speakers. And when you do that, one thing you do, and I think you've seen an example of that, is you take one of the old heroes or the absolute heroes in your field, someone who's been over the years really driving the cutting edge of the field forward and has made continuous contributions and that you've followed over the 20 years this meeting exists. And then, of course, you can also take your new heroes. And I'm absolutely thrilled that Aviv decided to come here. She's really doing absolutely spectacular work on that I think is an enormous inspiration for where actually molecular neuroscience has to go. And what her background is, she's a computational biologist to begin with. So she studied in Tel Aviv in an interdisciplinary program that combined computer science, physics, math, and biology. Then became a Bauer Fellow at Harvard and then went to the Broad Institute where she's now the chair of the Broad Institute. And initially she's really largely used computation to look at really complex forms of gene regulation, dynamics of gene expression with the computational power to find nodes in signaling networks that matter and to essentially let the data tell you what's there rather than you having some hypothesis and what should it be and then trying to press the gain and loss of function out of that factor that you decided to marry and then make it have some effect. But actually to look at the data and let the data inform you what is the most important in regulating a process. So she's really a pioneer in developing computational tools. She then also has built really a big branch of her own experimental science where they're doing really remarkable time-resolved gene expression analysis, proteomics, to look at how cells respond to stimuli, how stimuli change gene expression networks and thereby change function of cells. And much of this was done in cells from the immune system, but you can of course already imagine that this is actually fundamental to what we're trying to figure out in a developing nervous system on how a cell integrates many different signals from the outside, how it then changes gene expression pattern, thereby changes function of the cells and of the circuits. And the type of analyses that Aviv has really pioneered, looking at the level of cell populations, but also looking at the level of single cells, will, I think, really be the way forward in understanding how the brain develops and how circuits develop in the nervous system. That's why we felt that she would really be a fantastic person to have as a keynote speaker here. And so we're really thrilled that she accepted to come to a meeting that is sort of a little bit outside of your daily life. But I think it will be an inspiration for everyone who's here. So thanks very much for coming.
Thank you so much for a superbly lovely introduction. I should put a disclaimer up front, because otherwise I will be extremely nervous when the questions come up. I am not a neuroscientist, and I probably could not have followed most of the talks in the meeting, because this is really not my field. As Peter said, I'm a computational biologist by training. I study molecular systems, especially molecular circuits within cells, although increasingly in the last few years, the circuitry between cells. And our focus for the last decade or so really has been mostly cells from the immune system. But I did always think that the nervous system is not that unusual. I think we have three major distributed sensing systems in the body. There's the immune system, there's the nervous system, and there are the fibroblasts. So at the end of the day, it's probably going to be related to each other. No one told the body that you should, separate all these things from each other. Having said that, in the last few years, we've actually had some interesting adventures in neuroscience, mostly because of our increasing fascination with thinking again about what can we say about what I would say has been a 300-year-old project to map the cells from which multicellular organisms are made, but in particular to build a human cell atlas. And this project started with the discovery of cells when people had the microscope in their hands. And whenever technology advanced in biology, so has our ability to define what the cell types are and what their functions might be. And we know that the cell's identity has many different facets to it, starting with the well-defined types and then to states that change dynamically, dependence on their location, in absolute coordinates and gradients, but also just the neighborhood of other cells around them and the interactions with those cells. It dynamically shifts through transitions, especially in development, and there is an entire lineage for any cell in our body that delineates specifically the entire series of cell division events that have led to this cell from the zygote. And so it's been a long-term endeavor, and every time that a new technology came on the stage, there's been a dramatic shift with what we could do in this endeavor. And a new technology that really emerged in the last few years has been the ability to profile genomic profiles, and especially to profile the RNA content of individual cells, which I will call single-cell genomics or single-cell RNA-seq. And today we have in our hands, as a community, both a core technology involving specific molecular biology techniques to do this profiling. We have increasingly good methods to prepare samples, especially from large numbers of cells. We have a good and growing computational toolbox to infer from these profiles what might be going on. And the scale has really increased dramatically, especially in the last year, year and a half or so, such that today we can routinely analyze tens of thousands and even more cells in a single day with single-cell RNA-seq. And so the availability of these techniques has really led the community to start thinking about making a new or a refined version of a human cell atlas. But when we think about the application of these kinds of methods to neuronal diversity, there are some specific challenges. So the first challenge is that in the nervous system, this is also true in others, but it might be particularly true in the nervous system, it's not just that there is a large and unknown number of cell types, but that we have a good reason to expect that they would vary orders of magnitude from each other. So there would be very abundant cells, but there would also be important and very rare cell types. Second challenge is, and this is quite unique to the nervous system, it's one of the worst tissues to work with if what you want to recover is individual intact cells. And especially in adult brain, this becomes almost impossible, if not completely impossible. So if you want to do single-cell analysis and you can't recover single cells, that might be a problem. The third challenge is that some of the unique populations, even if they know that they exist, if we know that they exist, might require specific ways to capture them. One might want to think about, for example, neurogenesis and the availability of rarely dividing cells in the brain. And then finally, in neuroscience, more so than in other fields, there is an entire scheme of cell classification and description and taxonomy which is independent of molecular profiles, or at least has never been harmonized with them. It might depend on morphology, it could depend on the connectivity of cells to each other, and ultimately to the function that these cells perform. And very often, we might ask ourselves, would molecular profiles actually be congruent and harmonious with these alternative definitions that are attached with most of our legacy knowledge of how the system works? And so today, I'm going to describe to you our work to tackle some of these challenges, and I'm going to touch throughout the talks on three different methods. One is called Div.Seq, and it allows us to perform massively parallel single-cell RNA-seq. The second method, sNuc-Seq, allows us to avoid tissue dissociation by analyzing nuclei instead of cells. And finally, I'll describe Div.Seq, which we use in order to capture very, very rare and newly born neurons. And I'm going to show you the specific examples where we have applied this in the neuroscience realm, and also talk a little bit about the computational methods that we've developed in order to infer out the signals that we were most interested in. And I'm going to start with the gold standard of neuronal cell identity, which is the mouse retina, because a lot is known about the mouse retina. It goes back to Ramon y Cajal, which is an example of the invention of a new technology, in this case, the ability to stain cells, and the dramatic impact that the ability to stain had on how we define cell identity. And so we reasoned that this could be an excellent place to start, because especially as computational people, if we want to develop an algorithm and we want to apply to data and we want some answer at the end, we had better have a gold standard to compare to know whether we're on the right track or not. And we're going to use all the legacy knowledge of the retina for this purpose. And so our first step was to develop a massively parallel strategy for single-cell RNA-seq. This method is called Div.Seq. It was published more than over a year ago. I'll go through it very quickly. Imagine that you can dissociate a tissue, obtain your cells, suspend those cells in little droplets. These are water droplets in a medium of oil with surfactant that stabilizes them. So it's like reverse salad dressing with cells inside the droplets. And in the droplets, you also have barcoded beads, where every bead has has a unique barcode which characterizes that bead in many, 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 many different copies. And so that would give, once we blow up the cells inside those droplets, the RNA of those cells will be captured on the beads and will get barcoded by these unique barcodes. And then once it is actually barcoded, we can collect all of those beads together. We can either stash them somewhere and come back to them later, or we can grab a bunch of them and make just one RNA-seq library, but we know the original cell from which each of the RNA molecules and from which each of the reads actually came. And this is actually what gives us that scale that we can process, these tens of thousands of cells per day instead of having to laboriously analyze 100. And so our first proof of concept was to take whole retina, dissociate it. We did it across 7 different biological replicates. We obtained 44,808 cells. And then we used a computational method that I'm not going to elaborate on. We don't use it as much today anymore, in order to group the cells. What I do want to explain is how to read this plot. This is a nonlinear embedding called T-stochastic neighborhood embedding, where you can think of it as a PCA, but it is no longer linear as PCA is. And so these are 2 dimensions. Each of the points is a cell. And cells whose expression profile is similar in high-dimensional space are also going to be close to each other in this low-dimensionality embedding. So it retains these local distances between the cells. You can see that they're colored here. The color is derived by a computational algorithm that clusters the cells into clusters in a completely unsupervised way. And now we can take the cells in each of the clusters and we can ask whether there are molecular markers that characterize those cells. And given those molecular markers, we can say, oh, do they actually conform to what, say, somebody who actually understands something about the retina, such as Josh Sains, our collaborator, would consider to be independent cell types. And in this initial analysis, in fact, the 39 clusters matched quite well into the known cell types, and so we felt kind of good that we're not totally off track in these kinds of endeavors. And more specifically, if we look at the major classes that are known to exist in the retina, both of neurons and of glial cells, then we obtained roughly the correct proportions of the expected cell types. And in those cell types where we expect to have subtypes, then in some cases, like bipolar cells or amacrine cells, we get about half as many clusters as the known number of subtypes, and those clusters actually separate quite well relative to known markers. But also, if you look at a cell type like retinal ganglion cells, where there should be dozens of different subtypes, in fact, there was only 1 cluster. And that was not a surprise to us. This was because our analysis was completely unbiased, experimentally speaking. And so retinal ganglion cells might have many subtypes, but they are also the rarest subtype of the retina. Now, we just took whole retina. 70% is rods. And so 70% of the cells were rods. And so only few of the cells were retinal ganglion cells. And this was not enough to even capture all the subtypes, let alone identify them as distinguishable ones. And so after a good proof of concept, feeling that the method works and this should be successful, we said, well, we're not going to continuously spend 70% of everything on rods. Let's do a little bit of enrichment. And so the next step was to use transgenic lines in order to pre-enrich just for the broad cell classes. And what I'm going to focus on today is the first class that we went after, which are bipolar cells, with the first goal being reaching a complete and validated and full classification of all bipolar cells. So a brief introduction to bipolar cells that you probably know better than I do. Bipolar cells come in 2 major classes. There are cone bipolar cells and rod bipolar cells. And we also distinguish them functionally as off cells and on cells. The on cells are, the rod bipolar cell, the cone bipolar cells come in both on and off flavors. The rod bipolar cell, there's only 1 subtype like this, is an on cell. And even this distinction of what comes first, cone versus rod bipolar or on versus off, was not entirely fully resolved in the community at that point. Nevertheless, these are the best classified interneurons in the retina, so that would give us a ground truth. And you can immediately ask whether the classification is already complete. In 2009, people thought so, but in 2013, using EM and connectomics, people actually discovered a new rare subtype called XBC, so it immediately became clear, well, who knows, maybe there's another thing that's rare, and we didn't know was there. Also, we can ask, once we get transcriptional subtypes, would they actually match one-to-one and be congruent with the other ways in which these cells have been defined? These subtypes were not initially defined molecularly. They were defined by their morphology and by the level at which they laminate. So these are independent, a priori, of molecular information, and so we can peg whatever we find relative to this classification. That was the general idea. In addition, if we found, hypothetically, a new cluster of genes, we could ask, does it have a distinct morphology? And that would validate, when a new finding was made based on molecular information, that it actually represents something that a neuroscientist studying the retina would consider to be a subtype. And so what did we do? We started by dissociating retina. At this age, it's still dissociable. We started by dissociating retinas, but these were retinas now from transgenic mice that have, where cells have a VSX2 GFP reporter, and that is a marker of all bipolar cells, but also of Mueller glia. We collected the GFP positive cells. We performed Div.Seq. We collected about 28,000 cells after QC, 30-something thousand before. And we also did a smaller set of experiments using a high-depth technique, low throughput, called SNuc-Seq. We performed a set of analysis that I will describe in a moment in order to group the cells, and then finally we will validate what we found. I want to highlight 2 points in case anyone here is in kind of the single cell of neurons arena. The first is that we chose purposefully to sequence these cells quite shallowly, about less than 10,000 reads per cell. We got about 1,000 or so transcripts per cell and less than 900 distinct genes analyzed per cell. This was on purpose. We wanted to maximize number of cells relative to number of transcripts. This is not the capacity of the method. It can go much deeper. And we also compared to dramatically deeper coverage, 500 to 700,000 reads per cell, and to SNuc-Seq2 that recovers about 5 times more genes per cell. And while I won't belabor this, I'm happy to answer questions about this. We feel that at least in this system, shallow sequencing of a lot of cells trumps other strategies. Now we wanted to identify groups of cells that might be called types by clustering them together. The visualization here is exactly as I showed you before. Every dot is a cell. This is the same low dimensionality nonlinear embedding, and the cells are colored by clustering. But analytically, what we did is actually very distinct. We analyze here not this picture. The picture is just shown for you and for me to have a beautiful picture to look at, but actually a k-nearest neighbor graph that was constructed on the 68 significant principal components in this data. Before we moved to doing these single-cell analyses, we never saw more than 3 or 4 significant principal components in gene expression data. So it was always nice to see that there's so much more going on. And this was done on, as I said, almost 28,000 cells. We used a community detection algorithm, actually 2 of them. One is called Louvain, and the other is called InfoMap. They were both developed in the world of web and social networks about 10 years ago, and they're enormously effective at analyzing this kind of data, and they're also linear in the number of cells. So you can grow your number of cells substantially, and they would still run. We identified that the clusters are stable by using Bootstrap on the nodes, the edges, and the K, and then we visually inspect things on these t-SNE maps post hoc, just for both visual pleasure and to see that the distinctions actually seem to look good. And you can see that the 2 main algorithms perform basically indistinguishably. You can do it only with 1 batch and you get stable results, but also that earlier methods, both from our own group and from others, actually do not perform as well on the data. Now that we have the clusters in hand, we can actually look for markers by identifying genes that are differentially expressed between them. But even before that, by just going with those genes that are known to be important markers in the retina and using them a posteriori, post hoc, in order to assign names or tentative names to the clusters. And in many of these cases, you just look at the marker, it's expressed in mass cluster, and you can just say that's the type. we also have to take into account the fact that actually for some subtypes there aren't markers today, and there we use a process of elimination, including cell proportions, other related types, and so on. And what we end up with is a cluster name assignment. So we gave them names. That's convenient for us in order to talk about things, but these are just prospective names. We actually have to validate them. And the need for validation becomes much more apparent when we actually take those clusters and we organize them hierarchically into a dendrogram and start looking at them a little bit more closely. So doing this shows some interesting things. For example, remember the distinction between rod and cone bipolar cells and between on and off cells, and who has primacy in the process. Well, it turns out that on-cone bipolars are transcriptionally more similar to the off-cone bipolars than they are to the on-rod bipolars. So the first distinction is cone versus rod bipolars, and only the second distinction is on versus off within the cones. And that, to my neuroscience colleague, was an exciting finding. I did not know this was not resolved before. No, that's not to say it is easy to resolve. It was just, that's the thing when you're a foreigner to a field. You actually don't know what's an important question, and you don't know what's an important answer, and you can often just breeze through something that is tremendously important because it looks to you like, you didn't know, and you can fixate on something that is totally trivial. And that's very difficult, actually, for outsiders, so, bear with us. The second thing that we've noticed is that we could readily identify the transcriptional subtype for the XBC cells that already had a known marker, but we also saw that they actually relate in the dendrogram as a subtype of type 5. So this was reasonable. These cells are kind of weird in terms of their shape, but they do laminate close to 5. So that was a reasonable prediction. And then came the two things that we called the complete surprises. The first surprise was that there was a type 5a and a type 5b, which corresponded to the two well-known subtypes of type 5. There was the subtype XBC that we now will call 5d, but there was a fourth type 5 subtype, and that was not known at all. There was no suspect, so that was interesting. And also, type 1 was split across two clusters, clusters 7 and 9, and now you might remember that I told you that we checked very, very, very carefully that these clusters are robust, different clustering algorithms, and different Ks, and different this, and bootstraps here and right and left. And this is because once we get to clusters, we want to really believe that there are two clusters in the data. So maybe subtypes for type 1. And so all of this emphasized that we had better be able to validate whether these results are reasonable. And in order to do this, we used the following strategy. We took each of the clusters. We predicted molecular markers by using standard tests for differential expression, looking for genes that are specifically expressed in one cluster, but not in the other clusters. And now we can do different things depending on which subtype we're actually holding in our hand. We might have a subtype that has another known molecular marker. An example of that is rod bipolar cells. And so in those cases, we can do a double color FISH or FISH plus immunostaining, known markers plus new marker, and see that they actually co-express in the same cells with the same morphology and nowhere else. We can also, in another case, we might have a type like type 7 that has a GFP line. There might not actually be an extremely clear molecular marker, but apparently you can still see the cell expressing something specifically based on, a regulatory region that is active in the cell. And in those cases, we can use GFP together with FISH or together with immunostaining, again, new marker together with known morphology. You can also do RNA-seq in bulk or in single cells, and we've done both of those, although I won't show those. This would validate markers for known types. Now, there could also be more difficult cases when there are neither markers nor lines actually available for the type. And so in this case, what we have in our hand, and that's why we love the retina so much, is morphology and lamination. So how will we make this apparent? So we use sparse morphology labeling using lentiviral delivery. This is a system that was developed in Connie Sepko's lab, and FISH for additional markers. And now we're going to see whether the marker is actually expressed in the appropriate type. And I should point out by now that whenever I say this general we and the lines and the mice and the retina, that is not my lab. In case it wasn't clear, that's Josh and Connie. And so depending on the specific step. And so we have to start off with finding the markers. This is not rendered the best, but it's reasonably good. You can see that the different subtypes have different distinguishable markers. They don't always have as many. It has to do with how many others are like you and so on, but there's always plenty to go around. And we start with a simple case of the known type with the GFP line, in this case using road bipolar cells as an example. In this case, the known gene is PKC alpha. We add to it RNA fish for each of 25 different markers predicted by the clusters. And in each of these cases, the new road bipolar marker was validated by the two-color fish by co-staining together with the canonical marker. That was nice. Here's an example when we have a GFP line. So, for example, in type 7 cells, as I mentioned, there's already a GFP line. It's the Gustafson GFP line, and it labels well type 7 cells. And so we did fish for each of our new markers. Here we're showing IGF-1 for each of our new markers together with the, on retinas from the GFP line. And again, we found that the markers are completely congruent with the expression in type 7 cells. And we did similar validations for each of the other types for which there was an available line. But in many, many of the types, including, for example, type 4 or type 6, we currently don't really have markers. There are some that people debate in the literature, and in some cases there are simply none at all. And so in this case, the only definition we have is morphology and lamination. We use this sparse lentiviral labeling that I mentioned together with the fish to see if our new markers are expressed in the cells with the expected morphology. And the nice news for us is that in every single type, not just every single type we tested all of them. So in every single type, 3A, 3B, 4, 6, and 7, there was a matching, and we had a one-to-one morphological congruence, meaning whenever we see the marker expressed, it is expressed in cells with the appropriate morphology. There is no off morphology expression. So that validated all of those clusters. In addition, what I did not highlight so much is that one of the clusters, just one of the clusters, actually had additional substructure in it, and that structure broke to 2 subclusters, and those subclusters we suspected were type 8 and 9, for which there weren't good markers, so we couldn't definitively say they're 8 and 9, but then we went with this sparse labeling and with markers for BC9 or for BC8 and validated those as well. And so now we felt really good about all the stuff that's known, and we felt that those clusters are likely reasonable, and the markers are likely correct, and you can actually match morphological taxonomy with molecular taxonomy. What about the surprises? And so first, we looked at the type 5 cells, which are these 4 clusters, and let me remind you that we thought that there might be 4 subtypes in type 5, 5A, 5B, what we called 5C, and the XBC type that we renamed 5D. And so we went again and used the sparse labeling approach, and we were confirmed type 5A, and we were confirmed type 5B with the appropriate morphology. We also confirmed that the XBC type, which we now called 5D with our markers, indeed has the XBC morphology, so that was nice. But we also confirmed the presence of a new type, 5C, that has a very distinct morphology. It's called the tall morphology, and that was congruent with the molecular profile. And very nicely to us, in parallel to our work, which was entirely molecularly driven, Sebastian Sung and his team has actually done work with EM and connectomics and found direct evidence in the EM for the same morphology, the tall morphology, or as they called it, 5T. Now we turned to the 2 putative subtypes, type 1A and type 1B. Now there's a known marker for type 1. It's called mito-P. And so we took mito-P and we repeated mito-P co-fish together with distinct markers predicted by our clusters for 1A and 1B. And so we start with a marker for 1A, and we basically get a cell that looks like a bipolar cell and co-expresses mito-P. Great. We didn't really discover anything new yet. Then we went to type 1B, and something very interesting happened. We definitely got co-staining with mito-P. That was not a problem. The problem is that we have just discovered the unipolar bipolar cell. The cell only has 1 pole. It doesn't actually look like a bipolar cell at all. It looks like an amacrine cell. So that was interesting. And it was expressing the correct marker of a bipolar type 1 cell, except that mito-P is known to label type 1 cells. It was a good choice. But it's actually also known to label a very special subset of amacrine cells, which are NGNG. I always get stuck on this. Non-GABAergic, non-glutamergic cells. And so one simple possibility is that we have just found a great marker and a great cluster and a great contamination with amacrine cells, which would be fine. that's no foul. It's fine to find the contaminants as well. That's one of the benefits of single-cell analysis. You actually don't have to have pure populations. So we went one step further, and we went for a different marker, PPP1R17, because PPP1R17 is a pan-amacrine cell marker. If these unipolar cells are indeed amacrine cells, they should express PPP1R17. But type 1b cells don't express it. And on the other hand, they do express VSX2. They weren't just some contamination in the sorting for GFP-positive cells, and which is the bipolar canonical cell marker. And so we could find VSX2 plus cells that have an amacrine-like morphology and co-express the markers of the 1b cells. And so here at P17 sits a cell that looks morphologically like an amacrine cell and looks molecularly like a bipolar cell. Where does it come from? So when in doubt like that at the end point, go back in time and try to use developmental biology to tell you where you came from. So we started going back in developmental time, and as we got to P8 and subsequently to P6, we started seeing cells that were type B molecularly. They did not express any type 1A markers. They did express the bipolar cell markers, and they had 2 poles. And what ended up being the case is that earlier in development, this is a bipolar cell. And then gradually it shrinks one of the poles, it migrates a little bit lower, and it looks like an amacrine cell. And those cells sustain in this particular morphology and molecular profile at least until P100. So we think they're perfectly stable parts of the retina. And so this still leaves completely open the question of what is actually, driving this process. What's the code? That's something that for people like me is particularly fascinating. I mostly study regulatory circuitry that controls cells and their identities and their responses and so on. So for us, just knowing what the cells are is a starting point. It's not the end point. So we can take a brief look, for example, at how transcription factors are expressed. And what we see is not one transcription factor or even a set of transcription factor completely distinctive per type, but we most definitely see completely distinctive combinations per type. And this is something that for us is not entirely surprising. Hematopoiesis looks like this as well. So this might be a general principle, but it was compelling. Irene, the postdoc doing this work in Josh Sain's lab, actually did her PhD in the world of splicing, and an original goal was to find the splicing factors that are differentially expressed, and this figure would be called a bummer in that context, because nothing seems to be going on. Now, big caveat here. This is end sequencing. It quantifies the levels of things. It doesn't tell you there are splice variants. And splicing factors are well known to be regulated by alternative splicing that usually they actually execute on themselves. So there could still be a lot going on, but this would not be sufficient in order to find it. But it's not just their levels. And we don't think this is just a bipolar cell thing because we're quite well on our way to finishing the amacrine atlas, and in the amacrine cells we got exactly the same phenomenon. Very little going on in the levels of splicing factor, and the same type of seemingly combinatorial code in the amacrine cells, and in the bipolar cells we started hitting on the factors from the combinatorial codes, and we're seeing real functions there. So we think that this code is not totally off base. So to conclude this part, I showed you that single-cell RNA-seq distinguishes cell types very effectively. Large cell numbers, I didn't really go into all the geeky stuff around that, but large cell numbers are really critical when you have a complex tissue. The number of cells is probably the best thing you can do for yourselves, more so than the depth per cell. The computational methods are robust and efficient and scalable. You can really apply them to other things too. We found dozens of new markers, including for the established types that before did not have known markers. We found an informative classification of the on-off-road bipolar cells. 4 subtypes now exist for subtype BC5, 5A, 5B were known before. 5D is XBC, it now sits with its right friends, and 5C is a brand new one. And we also found the new subtype of type 1, type 1B, which has this unique unipolar morphology that emerges during development. We're working on completing the other subtypes. As I said, we're well on our way on the amacrine cells. We're now in really good shape also on the retinal ganglion cells. And in addition, we're doing a differentiation atlas along the time course and looking at the transcriptional code and doing cross-species analysis going all the way to human. So I hope I convinced you that you can harmonize with morphology, at least where that classification is extremely robust, well-defined, and experimentally tractable, and that you can really go after a system where the number of cell types is particularly large and varies many orders of magnitude. There's a way of actually going after these problems. What about the remaining two challenges of dissociation of dense tissue, especially in the adult brain, and enrichment for rare cells, in which, in this case, I'm going to talk about rare dividing cells. So in the remaining few minutes, I want to tell you a little bit about our recent work in this area. And I'm going to start with the question of the dissociation challenge. So we basically admitted defeat up front. We said there's never going to be a way in which we're going to get this gorgeous neuron with all of its lengthy, beautiful projections intact in our hands. No one seemed to know how to do that, and it seemed like a very difficult physical problem to solve. And so we said, let's not try to get cells, let's just try to get the nuclei. And if we could get the nuclei, we might have enough RNA in the nucleus to actually make things work sufficiently good, at least for some problems. And so the outcome of this was a protocol that's called sNuc-Seq, for single nucleus RNA-seq. And in this process, we dissect the tissue. We can take a very small piece of tissue, drop it into RNA later, or actually just snap freeze it. The published paper talks about RNA later, but you can freeze it all the same, maybe even better. And then we can isolate the nuclei by gradient centrifugation. And then, for example, we can sort them even using FACS before we perform single-cell RNA-seq. The nuclei are much more uniform than cell bodies. That's not that surprising. But what actually surprised us is that if you take the bulk nuclei of a tissue and the bulk tissue, there's very little difference in the RNA profiles that you see. That surprised us very much. We never thought this would be true. We never actually did the experiment until the reviewers suggested that it would be a good idea. And they were absolutely right. And so the other thing that surprised us is that in the brain, and now the Allen folks are reporting a similar result, kind of informally, that in the brain, single nucleus RNA-seq gives you the same or even better quality than single-cell RNA-seq, which is really single-cell body RNA-seq. And this is probably just because of the lengthy amount of time you spend, you know, in 37 degrees with a bunch of enzymes trying to break the thing apart. That's not very good for RNA. And it allows you to enrich for markers as long as they're nuclear. And so very briefly, we applied this to 4 regions in the hippocampus, and we easily recovered the major cell types. This is, again, a plot like the ones I showed you before. There are only 1,500 cells here or so, so far less dense than before. Each of the dots is a cell. They're close to each other if they're close in high-dimensional space, and the coloring is by a clustering method quite similar to the one I described before. And they easily fit later on with the regions we actually know we dissected them from. So that was a nice sanity check. The GABAergic cells were actually enriched with a specific marker, nuclear marker, so that shows that that works. But we immediately asked ourselves, you know, that's a cloud. Maybe there's additional structure there. And it turns out that recovering additional structure out of these clouds is not entirely trivial, largely because of a lot of noise in single-cell RNA-seq data. And so we devised a little algorithm that starts by the original steps of doing the dimensionality reduction and recovering of some image, such as this cartoon. And then we take each of the genes in our profiles, and we actually score how the expression of the gene. So the dark blue cells are now expressing the gene highly, and the light blue cells are expressing it lowly. And we're asking, well, are the cells that expressing this gene highly, they might not be separable into a distinctive cluster, but they might all be aggregating in some corner together. And if that is the case, then that's probably not a random thing. Whereas if the gene is kind of popping around all of them kind of all over the cloud, then that just might be noise. And that distinction is impossible to do before we actually know how the cells relate to each other. But after we do that, we can just filter and capture those genes with those more distinctive patterns, repeat the process, and after repeating the process, identify new clusters. Now, this is nice in principle, but what's particularly nice is that it actually works really well in practice. So, for example, applying it to the GABAergic neurons identifies these clusters, and we can find that very classical markers, neurotransmitters, cell surface molecules, and the like are actually expressed completely distinctively in those clusters, and then we can do the same tricks that we've done in the retina, the in situs and the fishes and the known markers and in order to actually validate these clusters. So that's nice. But sometimes patterns are not that discrete. You know that, again, way better than I do. I'm learning this just as I go along. You know, there's gradients, there's stuff. and so on. So here, we're kind of fortunate that in parallel to doing all this work, we're also really fascinated by, these spatial patterns. So for the past few years, we've been collaborating with Alex Shear's lab, who studies in this context, they study a lot of things, but in this context, early zebrafish embryos, and asked whether we could figure out where cells actually sit in space if we had a little bit of in situ data that somebody didn't put on some database years ago, and single-cell RNA-seq profile from the same developmental stage. I won't go through the details of the methods. That could take another 10 minutes. But the general idea is that on the one hand, you have your spatial, on the one hand, you have your spatial reference map. That comes from previously performed in situs with a minor amount of post-processing. So I can now say gene X expressed here, gene Y here, and gene Z here. On the other hand, you have your single-cell RNA-seq data. You do something to get rid of the noise. Now you can look for the cells that express X, Y, and Z, and you can say, well, those that express X alone should be here, and those that express Y alone should be here, and so on. And if they express a combination, there might be a place where the two intersect, and that's the right place. And so in this way, you actually end up projecting the cells into a location. I won't belabor, we validated this extensively. It turns out that it works really well. And so we applied the same approach, which we call Seurat, in the case of the brain. Again, we needed a database. Fortunately for us, the Allen Brain Atlas is exactly that kind of database. So we took the region that we analyzed, took the Allen Brain Atlas, made ourselves a little reference map, used a small number of landmark genes, and mapped all of the cells to their positions, now working actually cluster by cluster. And what we found is that the cells in those clusters, they're not really discrete clusters, but they actually map along these lengthy gradients, both dorsal, ventral, and medial lateral gradients. Great. So we can analyze brain tissue. This was adult mice, and actually some of them were really old mice. So as old as the tissue is, we can take care of it. We can even do post-mortem human brain. What about the rare dividing cells? That's the very last bit for today. And so this was one of those places where, the gods smile at you and you're just lucky. So it turns out that sNuc-Seq is extremely compatible with EDU labeling, such that after isolating the nuclei, you can just tag the nuclei with click-IT chemistry if they were already EDU labeled. How will they be EDU labeled? Well, we'll first pulse EDU in vivo, which is something people do all the time. It will incorporate into the proliferating cells. And then a time window afterwards, either short or long, we will capture those, we will take the cells, we will click-IT them, we will capture the nuclei, we will then use click-IT in order to label them, and then we'll isolate only the labeled nuclei. In this way, enriching for the cells that were originally EDU labeled. So we called this method Div.Seq, for a method to identify dividing cells, and we decided to apply it to our system. So we were studying the hippocampus, and in the hippocampus it is known that neurogenesis can occur in the adult, but that it's also not a very common occurrence. In fact, using signatures for what we thought were likely progenitors or recently newborn neurons and the like, in the entire data set of 1,600 cells or so, 4 seem to maybe have been newly born neurons in the original data set. So obviously we would have had to enrich a lot in order to see enough of them. And so what we did is a time course, at first in 2-day intervals, and in the end another additional week. So we did an interval of 1 to 2 days, 3 to 4, 5 to 6, 6 to 7, and then another 14-day time point after EDU labeling in each of these cases. And when you plot the cells in this 2-dimensional plot, you can see that they actually follow a trajectory, and there are ways also to recover those trajectories. Those are actually pioneered by some of our colleagues to recover this trajectory, where at the end, one end of this trajectory are the stem cell progenitors, and at the end, the newly mature neurons. And once you have the cells ordered like this, you can actually temporally put them in order, regardless of the time points in which you quantify them. So now time becomes this axis. In the field, this is called pseudo-time, because it's not real time. It's our understanding of biological time, of the progression of neurogenesis in this case. And then you can look at gene expression along this maturation trajectory and cluster genes into these dynamically changing clusters of gene expression, seeing which genes are expressed early and which genes are expressed late, and it's almost completely fluidly continuous, and you never had to synchronize anything, which is always very desirable. Now you can take the same method and you can say, well, I wonder what's happening in other parts of the mouse. So in our case, we decided to take a look at the spinal cord. We looked at the spinal cord because somebody already said that there might be something going on in the spinal cord, but there wasn't a definitive answer about this organ, so it seemed like a good place to look. At the top, you can see the EDU minus cells. There is basically no cells that could be considered immature neurons in the cells that we've analyzed in EDU negative cells after 1 week of labeling. So the animals were labeled, but these are the cells that are negative for EDU. But the same is true also if we just took the bulk of cells. There's no distinction. We can't see the immature neurons in this case. What we see is mature neurons and a little bit of the OPCs, oligodendrocyte progenitor cells. However, if we take the EDU plus cells from the same experiment, and almost 30% of them look like immature neurons, and in addition, we have this huge expansion of the OPCs to 54%. So we most definitely can hold in our hands what looks like newborn neurons, and we can repeat the same exercise. There are simply fewer cells here than in the hippocampal experiment, but again, going in 2-day intervals and going all the way to day 7, and also incorporating some of the non-EDU cells as a reference at the very tip. You can see the tip going from stem cell progenitor to the mature neurons, and you might see these 2 points that look like branching points. Our cell numbers are too little to really commit to this being something, but that might be alternative paths to gliogenesis instead of neurogenesis. And then finally, you can start comparing the programs. We did the same exercise in the olfactory bulb, where it is well known that there are newborn neurons, and you can see both a shared core maturation program across all 3 of the sites, and you can also see distinct profiles. Some of them are related to the distinct cell types, so we would see them, say, in the olfactory bulb and the spinal cord, but not in the hippocampus, in the dentagyrus. And some of them are related specifically to the location, so distinct between the 3 sites. And these results are very robust. And so to conclude this part, I showed you sNuc-Seq, which avoids the need for dissociation and lets us work with either lightly fixed or frozen tissue. It's applicable to any complex tissue, and we have tried it on many other things that don't succumb to dissociation. It works well for cell subtype classification, and we can find cell type specific expression patterns, and it actually compares to, and is sometimes much better than, single neuron RNA-seq. This does not generalize to other tissues that dissociate well. That's really because of the dissociation problem. And it lets us isolate and characterize real cells in the brain with nuclear markers. And the phenomena that we can see are pretty subtle. And then I told you also about Div.Seq, which is a high throughput and unbiased way to isolate and characterize rare dividing cells, or cells that have divided quite recently. It lets us monitor the dynamics of adult neurogenesis in the mouse hippocampus, in the dentagyrus. We also show that there are rare newborn neurons in the intact spinal cord. This is without any injury or anything. It was just a mouse. By the way, we have shown many of the results across multiple ages. I forgot to mention that. And we found that the neurogenesis programs, there is a core program, but there are also programs that are either cell type specific and shared between locations, or location specific and not shared between cell types. So together, Drop-Seq and sNuc-Seq and Div.Seq, we think are good applications for neuroscience problems. Not only for neuroscience problems, but also for neuroscience problems, and some of them quite specifically tailored for the realm of the neuroscience challenges. This does not mean that there are no challenges remaining, and that's good for people like me, and we like techniques and methods and so on, so we'd better find a new problem to solve. So there's a whole range of them. There's a problem of scale. No matter how many you can come up with, somebody wants 10 times more. That's true for all techniques, and they also want it 10 times cheaper, and then it costs them exactly the same because they need 10 times more for 10 times cheaper, and then they want another improvement. And so many cells, unbiased, any sample, and a very streamlined process that anyone can deploy anywhere would be highly desirable. There is a lot of work on that across many labs, and things are looking very rosy for the field in this respect. Getting multiple measurements in one cell of different facets of the molecules in that cell is very important. Measuring the DNA, the RNA, the protein, the interactions, and the interactions between cells, which in the nervous system is particularly critical, are all things that are on the horizon. There are proof of concept experiments demonstrated and published for many of the pairwise combinations. There are starting to be three-word combinations, and a lot of the problems will be to scale this, to make this robust, and work for other things. But this is where single-cell RNA-seq was 3 years ago, and so I think in general the community does not feel that this is an insurmountable barrier. And that would leave a lot of room for maneuver. Spatial resolution, obviously, is a big one. There are increasingly good methods for multiplex in situ measurements in tissue, and up to hundreds and maybe thousands of transcripts. There are methods for inference, like the early ones that I described to you, that also, again, multiple groups work on. And this also opens the way to newly defined cells simultaneously based on their intrinsic profiles and the profiles of their neighbors, which is something that is a very interesting algorithmic problem. And then finally, I think it is critical to always think about function. The reason our lab went to work on immune cells is that in immune cells, you could do all this molecular stuff and genomic stuff and predictive stuff, and then you could actually go and stick the cell back into an animal and see what happened. And I think in neuroscience, it's the same. That's my outsider's view of neuroscience. If there isn't a function at the end of the road, it's a story, but it's very hard to do much with that story. And so single cell profiling is actually compatible with a lot of strategies for perturbation, for lineage tracing, all sorts of things that one would want to apply in the animal context. But now you would have all of that information, and for every cell where you have this information, you will also know its molecular state from its molecular profile. So I think that's a very important and really up-and-coming area of investigation. And then finally, I think the adventures we and others have had in neuroscience in the last couple of years, or in analyzing neural systems, I wouldn't call this neuroscience just yet, in analyzing neural systems have actually provided us with really important lessons in our general endeavor of my community to think about how to build a human cell atlas, which is really something that's becoming increasingly reality. How to think about healthy tissue, what to do with disease, where is the place of animal models versus human studies, where do you get samples from, which technologies are useful, what computational approaches do you need to apply, but most importantly from all of those things is how to engage with a biological community that actually knows something and cares something about that system. It's very nice to have a lot of tools at your disposal, but you would never develop the right tools and you would never deploy them well if you don't collaborate with people who actually care about that system, with the entire community that works on that system. And I think that is a very good place to stop and also to thank my many collaborators. And so the work that I showed you has been several chapters of work for our lab. It started with Div.Seq, which was a technique development project, which was led by Evan McCosco, a phenomenal postdoc in Steve McCarroll's lab, who's just now starting his own lab at Harvard, and Oni Basu, who is a physicist, soft matter physics, and a device whiz. She developed all the Div.Seq devices, and she was a joint postdoc between my lab and Dave Weitz's lab, and is now starting her own lab in the University of Chicago. And the analytical work for that project was mostly done by Rahul Satija, who I think is missing from this slide and only present on the next one, who is now in the New York Genome Center in NYU. The work on the retina, you know, I could not be more thankful for Josh Sains to kind of chasing us and telling us the retina is a really good system, you should really try it out. And explaining to us why it's so good and how much we could learn and how we would get really meaningful answers from working in the retina. And then also chasing Connie Sepko and telling her that she should really join forces with us as well. And so it's been a wonderful three-lab collaboration between Josh's lab, Connie's lab, and my lab. And it was led by three postdocs, Sylvain Lapan, who is a postdoc in Connie's lab, Irene Whitney in Josh's lab, and Karthik Shekhar, who's a computational scientist in my lab. And they really did the bulk of the work that was shown, together with Evan McCosco and Steve McCarroll, who remain our partners in crime. And then finally, the work on sNuc-Seq and Div.Seq is part of a long-term collaboration between my lab and Feng Zhang's lab at the Broad Institute. It was led by Naomi Habib, who's a joint postdoc between both of our labs, and Yinxing Li, he just graduated, a grad student in Feng's lab. And there were some critical contributions, especially when we had to do a very large number of experiments in a short period of time by Inbal Davidi Avrahami, a postdoc in my lab, and Lukas Schweich, who is a and Matthias, who are postdocs in Feng's lab. And I want to leave you with this gorgeous picture that Irene drew of herself. That's actually her from the back. Thinking a little bit about how, you know, these three very different views maybe of the world are actually coherent, congruent with each other, and might tell us something useful about neurobiology. Thank you very much.
Thank you for a spectacular talk. There is, who's the first with the microphone? Here, go ahead.
Okay, very impressive. I was wondering about the number of genes or gene expressions that you need to actually make the distinct population. So you said something about deeper sequencing and not so deep, and so I was wondering in the example of the bipolar cells. So if you want to distinguish them all, how many genes do you need to know, and where do they fall in terms of groups? So they could be coming from transcription factors, surface molecules, and so that you have a minimal set. Yeah. And if you go above, you don't make much difference, and when you go below, you lose the distinction.
Yeah. So let me make two distinctions. There is the number of genes that actually distinguishes one cell type from the rest, which varies. Some cell types are just more distinctive, so you will see more genes. And there's the number of genes you need in order to make the distinction, which can be a far smaller number and is far less critical. The number of genes in the retina system, in the bipolar cells, which seem to be actually a really low bar. When we went after them, we thought we were setting ourselves to a substantial challenge, 100 subtypes and this and that. But it seems to be that looking at the data, first of all, discretely, you don't even need real quantitation of levels, and a relatively small number of markers. I would say you could do quite well with 10, 15 per cell type, you would be fine. But that's the retina. That would not necessarily generalize to other things, and that's only for typing, which is only one aspect of what makes a cell a cell. Which classes? That's a question we actually did not systematically look at. What I can tell you is that and you saw this actually with the transcription factors, what I showed you for the transcription factors were the most specific ones that we could find. And they weren't as specific as the markers I showed you. They weren't even close. The markers were like expressed only in one type and pretty dead in all the others. The cell types always, the transcription factors always oozed about. And that's what we also know from hematopoiesis, which is a system I've studied for many, many years extensively, and so have other labs. In hematopoiesis, people for many years had the feeling that there are these master regulators, and they are very lineage specific. And it's true that if you manipulate them, bad things happen to lineages. But it is also true that they are kind of shared all over the place, and they combinatorially combine into these modules that cross boundaries. Whereas you have beautiful markers, you know, say CD3 and CD4 and CD8 are amazing markers in the realm of T cells. You can't mimic that with transcription factors. So I think transcription factors would be underrepresented in this class of markers, quite definitively.
Actually, I have a follow-up question on the bipolar, and also you mentioned briefly about amacrine cells with your combinatorial transcription factors, but not so much in the splicing factor.
Yeah.
To this audience, I think we are all very interested in what will cell surface molecules, when you analyze them, what do they look like? So
in fact, like at 12:55, I was still debating between two versions of this slide, which I once did with adhesion molecules and one without, and chose the one without. The adhesion molecules look quite similar to the transcription factors. And I actually added that in Cold Spring Harbor 3 weeks ago, well, not Cold Spring Harbor strictly, in a Banbury meeting I was at 3 weeks ago, because somebody was saying something similar about adhesion molecules in a different system, and I wanted to show it. It was one of those live meetings, right, where you kind of can show things in real time. And so, and that is actually in the published paper too. It shows that, so you can look at the picture and see. But it looks much more similar to the transcription factor gestalt than to the splicing factor, but also importantly, it again does not just look as every type has its own.
Not like a PT.
But there is sufficient numbers there that would fall into the categorizing genes. Yeah. So there may be in between is a good place to put them.
So I'm interested in the spinal cord, if you give me some more details. What was the age of the spinal cord that you did your Div.Seq on?
So these were adult mice, strictly adult, not young adults even, but I do not remember the exact age off the top of my head. Okay. I could check like right after.
I may ask you.
Yeah.
And
so yeah, this was most definitely adult as anyone would define. I don't think the spinal cord experiments were in really old mice. The dentogyrus experiments, we spanned all ages, starting from young to adult to old.
And was there anything in the way that you dissected the tissue that could tell what layer of the spinal cord
Not that I would know, but that's my ignorance, not the experimentalist's ignorance. I didn't even know that was a question to ask. Yeah. Well, you know, if I can't admit my ignorance, I'm in deep, deep trouble in what I do. So, yeah, but it's an excellent question, and it's one that Naomi could answer immediately, right? Her hands dissected. So I just don't know. Yeah.
I wonder what these...
I have no idea. Yeah, thank you.
I wonder what these clusters or cell types mean in terms of biological networks. Could these be different steady states of the network, or is the network randomly sampling different expression pattern and we are just picking up at different time points?
You mean the well-defined types?
Yeah.
The well-defined types are stable states of if you think from transcriptional circuitry, they're probably stable states of the circuitry in a pretty robust chromatin landscape. That would be my guess in the retina, not my knowledge in the retina, but it's based on everything else we've ever looked at as a community in terms of transcriptional circuitry. This is not the end of the state space that retinal neurons span, because this experiment is without any stimulation. And one interesting question to ask, and there are still these clouds within them, right? Mm-hmm. So one question to ask, for example, is if you did a directed experiment with stimulations and you compared on top of the cell type, what are the states that the cells assume? So what we know from many other systems, including in the nervous system, right, is that you might have your type of cell, but that type of cell receives a stimulus. It's also going to be transcriptionally responsive to that. And so there's going to be movement around this core type in terms of the state. And that's perfectly fine. It's just that this experiment has not yet looked into that at all.
One more question. How difficult is it to reconstruct the network from this data set? Can I infer the regulatory relationships from this data?
So you can start doing very simple types of inferences that we've done a fair bit of in the past. That used to be my main profession. So you can look at, for example, what works really well is if you couple a hierarchical model of how the transcription factors are expressed across the type and try to predict with it how the rest of the genes are expressed, it will probably recover most of the important factors. Our specific focus turned a little bit into the developmental path leading to this, so that we would have also temporal information to harness rather than only doing it at the endpoints. So you can infer quite a lot from the endpoints, but there might be things that are lost to you by that. Transient events that are critical for specifying the lineage but are not stably maintained. So you can probably go quite a long way. We did a little bit. We didn't end up actually publishing it in the paper, mostly because we, by that point, knew that we could do better, and so we didn't want to write something that's kind of half-baked around that. But the signal from these things, variation in general is just phenomenally good for constructing transcriptional relationships. So any form of variation. So even noise between cells, not technical noise, but biological noise between cells is good for finding regulatory relations. So no doubt you can. Yeah.
Thank you.
Yes.
Hi, I was curious, have you tried combining lineage tracing to define the end states in the Div.Seq maps?
So we have not. We have not. We work extensively on coupling all sorts of genetic information together with these types of readouts. We have, our main focus initially was in something else that's related to the immune system, and we're kind of wrapping that now. So the thing that we're focusing, this is one type of application. And now there are tools that have emerged very recently for lineage tracing, which are very cool. Most effort in lineage tracing is focused on leaving the trace in the DNA. And so you might want to think about ways of getting the trace to be expressed, and it's not undoable at all, and it takes some tech dev.
Is sNuc-Seq compatible with the...
Yes, and sNuc-Seq is compatible with DNA and RNA, which we are doing already. Yes, totally compatible with DNA and RNA. It's nuclei, and they're fixed. It's compatible with a lot of things as a result. In fact, most of the 1+ and RNA plus something methods work really hard because the something they add to is almost always around the genome. It's the DNA, or it's the chromatin accessibility, like attack, or it's methylation, any of those things. So it actually works easier. It works in nuclei, it works in fixed, and so on. The RNA is always the difficult partner in the marriage. So moving into the nucleus actually simplifies that a great deal. You're totally on track, and there's no doubt that multiple papers from multiple groups will come with many variants of this kind of idea, because every variant requires a fair amount of optimization and design and demonstration and so on. It's nice because there's room for a lot of activity. And this field is very open. The single cell field is very open. The labs talk to each other a lot, so people kind of know that there's a lot brewing.
Hi, here.
So I have two questions. First one is more about clarification. So does all this newly identified marker that exhibit binary expression in distinguished classes? Is that right? Or do they exhibit a continual expression pattern?
So in the Retina system, even though the data itself is not binary, you don't really need that. So if we took the data, which was quantitative, and we discretized it, we binarized it, not just discretized it, we did it 0, 1, you could pretty much recover the whole thing. That is not true for all systems. That seems to be the case in this system. There's sufficient stuff that's specific to every cell type and not expressed in any of the others. And we have our lab that studies a lot immune cells. I can tell you, in immune cells, this would not fly. It would not fly. Because immune cells maintain, especially at the bottom of their differentiation tree, a huge amount of fluidity, and that fluidity is all quantitative. Yeah. And it might not be true for many other pieces of the nervous system. It just seems to be the case in bipolar cells and probably in the other subtypes as well.
And the second question is about the depth coverage.
Yes.
So all this clustering is based on, it seems that pretty much affected by the highly abundantly expressed genes. And if you could able to increase the coverage ever, if you use a different classes gene would they basically regroup your subclasses?
So I invited that question, actually, right? I told you, ask me if you want to know about coverage. So we thought that that was a possibility, too. And instead of, some people have opinions and they just stick by their opinions, and our approach was, we have no idea, let's just try it both ways. So we did actually 3 different types of analyses with 3 different types of data coming from 2 experiments. One experiment is the main one I described to you. It was a drop-seq experiment, and it was sequenced shallowly. The second experiment was the same drop-seq experiment, but we sequenced it a lot more. And the first thing that we showed, and that's mostly important for anyone who actually uses these methods, but for the others you can ignore, the first thing that we showed is that there was sufficient complexity in the library. So if you sequence deeper, you actually recovered more genes and so on, and it looked really good. So that was technically nice to know. You can always go and sequence more, but it's very hard to go back and recover the sample. And the third experiment that we did is that in a subset of the bipolar cells, we actually went all out. We did our old traditional plate protocol, fact sorting the cells, doing single-cell RNA-seq with a modified version of SMART-seq2 that we ourselves modified to be extremely sensitive and recover large numbers of genes, and sequenced it extremely deeply. So we had 2 aspects. We had an experiment with fewer cells and tons of reads, and an experiment with many more cells and far fewer reads, and a subset of the big experiment, which was bigger than the really small experiment that had kind of an intermediate depth. And the answer in this system, and I'm saying that very explicitly, we cannot generalize anything except for this system. The answer in this system was that the experiment with the extra depth didn't give us anything extra. It gave us a lot less. It couldn't find anything more fine, and any distinction that it made was fully congruent with the distinctions of the shallow but big, large number of cells experiment. So whenever it made a distinction, it was congruent, but many of the actual distinctions that are there, including known types and new discoveries, it simply couldn't make because there weren't enough cells. So this is not that this is the best way to think sometimes as a scientist, but sometimes it's required. You can actually, at the end of the day, put a dollar amount on this. Imagine you have a fixed dollar amount. In our case, the Brain Grant Initiative gave us some dollars for the year, right? And that's all we can do at the end of the day in terms of sequencing and processing cells and the plates and the amount of time on the flow sorter. All of that becomes a certain amount. If we want to maximize the amount of biology we get out of the dollars, we better get more cells and more reads. That's what we learned. So that was the bottom line. But it was not, and it's not necessarily true, and it depends on the question you want to answer. We answered the question on cell type classification. It might be very different if we looked at questions about splicing. If we wanted to answer important questions in splicing, we shouldn't be doing 3' end sequencing. We should be doing full-length sequencing, and you can't yet do it in droplets. And then we would have changed our strategy completely. And when we analyze tumors, which our lab actually does a fair bit of, and we want to read genetic mutations, we do full-length sequencing because we need the mutations. So it's really a matter of the goal, but in this particular context and particular goal, there is no doubt what the answer was. Yes?
I'm wondering with your larger, shallower data set.
Oh, are you okay?
I'm wondering if with your larger, shallower data set, whether you estimated how many cells you would have needed in order to get the same functional segregation of groups. So did you have too many, or could you have done it with half as many? How many would you have needed?
Yeah, we actually had too many. We did have too many, and we did that. We did downsampling of the system in order to show, gradually going down and finding the inflection point at which you really start losing signal. But we had at least twice too many as we needed, even for the rarer subtypes. When you're early on in these types of approaches, you want to overshoot so that you can define where you should actually sit. So we overshoot on a lot of things. In many experiments, when we do them for the first time, we overshoot the sequencing, we overshoot the number of cells. So that was a distinction, and this is well reported in the paper, including all the curves and so on. The other thing that's worthwhile knowing is that you can actually use a very simple mathematical formulation based on basic principles like the central limit theorem to answer questions such as Rahul Satija, actually, when he was still a postdoc, that was one of the last calculations he did before he left the lab, was if you have a system where you have a rough guess on the number of cells, you don't have to be extremely precise, but on the number of different cell types that you have, and the proportion of the rarest type, that's the second parameter, and your desired number of cells that you would want to recover from this type, which for these kinds of differential expression analysis, we now know should be in the couple hundred cells maybe with these kinds of techniques, but you probably don't need much more, then you can actually calculate roughly how many cells you should be profiling to achieve that target. You will be overshooting on the abundant classes, but you will recover your rarer classes with a certain amount of confidence, and you can determine also, of course, your confidence interval. So there's an analytic way around it, and that's nice because then you can compare the analytic solution to what you actually observed empirically and have an idea whether the assumptions in the model, in the analytical model, are actually correct.
I think we have 3 more quick questions that we already have committed to. One is there, and
I'll
then our future organizer will have the last word.
Okay, so I think this might
be the person, thank you.
This might be a bit of a silly question, but you have this problem with 0 reads. So a cell might be expressing genes and you get a 0 reads.
I wanted to answer that on the previous question.
So how doesn't that affect these beautiful clusters that you
get? I was actually going to answer that when the person over there asked me the question and forgot to point it out. Because when I was asked that question, I was told, and you only recover the highly expressed genes. And that's not technically correct. The probability of detecting a gene is affected by the level of expression of the gene. That is absolutely correct. But you don't only recover the highly expressed gene. Your likelihood of having 0 instead of a number is much higher the lower the gene is expressed. And in fact, this is affected by the level of expression of a gene as well as by what we call the quality of the cell, which encompasses in it a lot of things. Most of them happened before molecular biology ever hit. Some cell types slice better, are bigger, are smaller, or this or that. We don't fully understand what it means, but we know that some cells give a better or worse library. So this 0 inflation, however, conditional on expression level, is actually a random event. And that is actually a very good thing for us. And if you remember those plots where I said, and the dots are kind of all over the place versus aggregated in one place, what I was actually assuming is that all over the place means there might be all sorts of zeros that also shouldn't be there, whereas aggregated in one place is a real signal. So the fact that we can assume, and nothing that we've and others have seen so far suggests that this assumption is horribly wrong, that we can assume that conditional on expression level, the probability of 0 inflation is otherwise random, lets us see a lot. But what really lets us see a lot, and why these methods actually work so well, is that there is co-regulation in cells. And so imagine that you have a set of 100 genes in a regulome, and they're all going to be co-expressed in this cell type. And it's true that you're going to miss out on a certain number of them. And it's true that they all might be relatively lowly expressed. And so they're all going to be dinged to some extent. As a set, you're actually going to see them quite well. So many times we actually score signatures instead of scoring individual genes. Or you can use imputation. You can use the data itself to identify this covariance structure, and then you can impute what likely are the behaviors. We try to avoid imputation as much as we can because it has a whole can of worms associated with it. But this covariation, this dependency means that even though you drop out, is the other name for these 0 inflated values, even though all these traits are there, there is a lot of structure that remains in the data. And most algorithms these days, in fact, they're underutilized in biology But they assume data that's sparse, modular, and noisy. So they're actually crafted quite well to handle all of these liabilities, and we're more and more going and picking up methods that people have not used in biology before, but besides that are actually quite well developed in applied math, in computer science, and so on, that just do well with data that has these types of behaviors.
Yeah. So that means this method isn't so good if you're looking for a particular gene in this data.
So for knowing the expression of that one gene in that one cell is going to be difficult. But ascertaining the expression of that gene in that cluster is going to be easy. So if it is in singularity for this one cell, you're not in good shape. But if that gene is co-expressed with a whole bunch of others, you can guess what its correct expression is. And in fact, in that zebrafish study, that's what we did. We took the original noisy study. We had 37 genes that we had in situ data for, so we really wanted to know them precisely, and we guessed what their expression is based on the expression of everything else that co-varies with them. Yeah. Yes?
Hi. So my question is regarding your different subtypes of bipolar cells that you've identified. So do you think there are further subcategories of transcriptomes within any of those clusters? And if so, are the computational algorithms that you've developed able to identify those differences within any of those subcategories?
In the bipolar cells.
Right.
So in the bipolar cells, we tried and could not find evidence for additional substructure at this level of resolution, besides the distinction between what we call between type 8 and type 9. They're known as type 8 and type 9. They were a clear structure of two subclusters there. We couldn't find it for other clusters. The methods, the algorithms can be applied in modes that would give you a far larger number of clusters, but you wouldn't find sufficient number of differentially expressed genes actually supporting those distinctions. You can always push an algorithm to give you more. What you ask it to deliver to you is what it would deliver back. You ask it for clusters, you'll get clusters, even if the data does not have clusters at all. You ask it for more clusters, you will get more clusters, even if there aren't more clusters. So you have to have something post hoc to actually convince yourself that what it gave you is correct. So in the bipolar, no. The reason I actually emphasized that point when we went to the dentagyrus is that there we also had blobs of things. And there, when we tried to push one step further, the blobs did separate. What they didn't do is in the retina. So we don't think in the retina there is more. We can never be sure. Somebody can be more clever, apply a different method, get more data. What could absolutely exist is a subtype that's below a certain level of rarity. But we can say what that level is. This is like human genetic variation. You can say, at the level I did this genetic study in the human population, I should recover variance up to 1%. And you know that things below 1% you simply won't see. So that's our goal, to put these bounds, rather than to say we found everything. Yeah.
Super exciting.
Thank you.
So I have a very practical question. So your lab and several other labs have published data online. So I was wondering whether, in addition to that, are there any other resources that those are already publicized and any advice of how to use them? So this could be very useful for the many smart labs who don't have the money yet or don't have a good friend at Broad Institute yet. So maybe we can look at something quick, some quick advice.
So, we live off of the same NIH grants, just so it's clear. So in terms of resources, I'll highlight one that already exists and one that actually under the umbrella of the BRAIN initiative, our group, a group in Berkeley, and a couple of others are trying to just put forth. And in fact, the NIH didn't really give us any money to do it, but we all feel it needs to be done, and we kind of cobble it together from nothing. At least you don't have to do any expensive experiments in order to do that, so that's nice. The first is what we call the single-cell portal, and it's just a place where you can post this kind of data. You can post it publicly, but we can also post it in a closed way, so you can use the portal and all of its tools in your own research, and when you're ready to release it to the world, you can, uncheck a checkbox and everyone can see it, or you can share it with 7 collaborators and not let anyone else see. So it gives you a lot of flexibility in this way. Our lab's data will all be, it already has, it has the sNuc-Seq data. It was actually developed. Naomi, the postdoc who did the sNuc-Seq work, was just willing to be the alpha tester and guinea pig, so her data got there first. And the bipolar data will be out very, very soon as Karthik is back from his travels. But after that, all of our data, including our legacy data that we already published since 2013, is all going to go there, and presumably other brain grant data will get there too, and we hope people will just use it. Open source, you can do whatever you want with it, including replicate it for your own research. The other effort that we're doing together with these other groups is to try and aggregate some of the methods so that people can run them, especially can run them on the cloud, because especially as these numbers of cells grows, you're getting into a place where you don't have the compute resources locally to actually analyze. And even if you do, starting to download certain data becomes a real issue. And we anticipate that this will grow as a problem dramatically in the next 2 to 3 years. So we're trying to preempt that. That's been going, not as fast as it should, blah, blah, blah. But I think we'll be fine. We've released almost every tool and method we've developed. Sometimes nicer and sometimes not as nice, and that a little bit depends on where, if the grad student already graduated, it's harder. And if they're still around, it's easier to wrap up the code and stuff like that. The usual issues. But everything almost is. So for example, the bipolar cell analysis, there's an R markdown, which means you can literally take your data and feed it in like boom, get the same results. And so I think that's the way that we want to go. The Seurat original method, and actually is a more general package that also includes the methods that we did in the first Retina paper, was also released and is now maintained by Rahul Satija because he moved to his own lab and has been used by a lot of people. So, and Trinity, which is our RNA-seq assembler, has a whole suite of tools for single-cell RNA-seq.
Thank you. Thank you.
Yeah.
Okay, I think with that we
That was
close. Thanks for a fantastic talk.
Thank you very much.
Thank you.